बस ये शुरू ही होने वाली है। आजकल बस ऐसे ही है, रोज का सुबह कुछ ना कुछ
Welcome back. You're watching India Today. Let's get you details now of the big headline from Prime Minister Narendra Modi when he spoke at a BJP event today. Addressing BJP workers, Prime Minister Modi, in a sense, seemed to want to settle the entire language debate and put to rest all the criticism that the BJP constantly faces of being a Hindi-following party. Now, what the Prime Minister has said in his address is that every language is unique and every language is important and equal. Very importantly also, Prime Minister Modi, they're saying that the BJP supports all languages. Let's take this across now to Kicha Sudeep, Kannada superstar, who's joining us on this broadcast. Sudeep, thank you for joining.
You just cannot generalize and say that everybody treats everyone badly. So when I said it, it's in the context because when films are coming from the South, they are considered as pan-India because we are going towards Hindi also. But then see, that's when I said that, it is not a national language where you have to consider that as one motherboard or the central focal point and everything is pan-packed.
ये भी ऑन है सारे।
हाँ भाई यही है ना,
ये हमारा
ये तो स्वैप हुआ है।
हमारा नहीं
ये कौन
हमारा
join-
वो है ऊपर
ये कौन ज्वाइन किया है?
लिखा हुआ है ना ये डीजी और
मैं वही सोचा ये तो यार इतना ज्यादा जूम हो गया है।
ये सिंगल आदमी होगा
ये आप
पूरा
आगे
टीम बैठेगी ना
सर, आप इधर बैठे थे ना आगे।
वो रिमोट भी
रिमोट
यहाँ पड़ा था कहाँ है?
इसका नाम क्या है? ऐसे रेडियो टीचर रखा है ये।
हाँ हाँ
हाँ ना, ये सीधा ही रख दो पहले जैसे था। ऐसा हो गया ना, इसको और सीधा।
अरे ऐसे
हाँ
सर इधर वाले भी तो चाहिए सर।
अब हुआ सर वो सेंटर नहीं है कमरे का
सर वो कैमरा सेंटर
केवल सेंटर पे नहीं है तो वो देखो ना इधर उधर दरवाजे की वजह से ये थोड़ा सा इधर है स्क्रीन बनना नहीं आता ये इधर स्लाइड कर
नहीं, जैसे पहले किया था उसको, इसको सेंटर में ले आओ, उस स्क्रीन के सेंटर में। अब ये इस जो स्टार्टिंग में था,
शर्मा जी ऐसे हमारे तो और करीब और आपकी फोटो नहीं आ रही
नहीं हम यहाँ से उठ के वहाँ जाएंगे,
और करनी थोड़ा सा
हम वहाँ जाएंगे ना
थोड़ा
ये। हाँ ये ठीक है,
इतना ठीक है
इतना ठीक
हाँ,
है,
इतना
इतना
ठीक
ठीक
है
है।
बस इतना ठीक
लेकिन ये अभी ये लेफ्ट राइट वो है, लेफ्ट
सर वो
राइट।
पोज नहीं अभी ठीक है
हाँ ना, लेफ्ट राइट ठीक है ये।
अभी ठीक है
हाँ ना, लेफ्ट
सर
राइट।
वो लिखे में तो उल्टा ही आएगा हमेशा आपको उल्टा दिखेगा
सामने वाले को सीधा दिख रहा
वहां
है।
पे जहाँ ट्रांसमिट हो रहा है ना
हाँ
वहाँ सीधा दिखेगा
वहाँ सीधा दिख
वो
रहा है।
जाके देखना
आपकी स्क्रीन नहीं आप
वो देख रहा है।
बाकी लोग तो खोलेंगे ना
सर
उनसे पूछेंगे।
सर
वो
लास्ट टाइम
चेक
हमने
करके
टेस्ट कर लिया था।
वो चेक करके
लास्ट
किया?
टाइम हमने
हाँ।
टेस्ट कर लिया था, सीधा दिखता है।
हमारी स्क्रीन का उल्टा दिखेगा बाकी सबकी स्क्रीन में सीधा दिखेगा
ओके।
हाँ
वो कर चुके हो
जैसे शीशे का होता है ना,
ये मेरे इमेज है ना
और एक दर्जन है यहाँ।
सर ये काली कुर्सियों की वजह से मच्छर बहुत ज्यादा होता
ये देखो कितने सारे हैं।
सर वो कितना
है
वो
है?
जूते भी नहीं पहन के आए हैं।
सर ये पहले नहीं होते थे ये ब्लैक चेयर की वजह से होने लगते
क्यों?
ब्लैक पे तो
ब्लैक डार्क पे आते हैं ना
तो कुछ स्प्रे पे करवा देना चाहिए यार।
सर रोज कुछ न वो
सर,
आगे
क्या
चढ़ाते हैं सर।
ये बड़ी दिक्कत है
ना ना ये बहुत सारे हैं, सब सारे डेंगू पंगु है।
ऑफिस में भी वैसे घूंघट
मैं वहाँ जा करके आता हूँ। पीके मिश्रा विकास, अरे नहीं सर कितने लोगों ने ज्वाइन किया भाई?
नीचे लिखा
नीचे लिखा है
ये हमारे
यार।
हमारे लिंक के थ्रू किया जाना है।
हाँ हाँ
100 लोग ज्वाइन कर सकते हैं। वॉइस ऑन है?
हाँ सर वो बंद कर देना
एक ये चीज बताइए पर जानकारी के लिए।
सर ये टॉवल चाहिए, ये टॉवल हट रहा है, मेरे पास तो जो लगा दिए एक टॉवल हट जाएगा। और क्या कर लेंगे, मदन मेरे पास है किसी के पास लगाना है, तो टॉवल ऑफिस में है जी, उसी को बोले जी, बोले सर आ जाएगा, आ जाएगा ठीक है, सर किसी और से पूछ ले यार, अरे किसी का चेयर से निकालना है,
किधर है वो पीपीसी?
विजय सबको म्यूट कर दो।
ये रखो पोजीशन, ये ठीक है, दिस इज परफेक्ट।
विजय सबको म्यूट करके रखो अभी टिल 11 o'clock.
Let us improve the utilization
Madam?
of this PV.
Natasha,
विजय सबको म्यूट करके रखो अभी टिल 11 o'clock. मैडम अब बोलिए।
Good morning, sir.
Good morning.
Good morning, welcome, sir.
ये बंद करना पड़ेगा
Thank you all.
सिस्टम से
मैंने सर के लिए ऑन किया था
मैडम इसको फोन पे मत खोलो।
अरे
Sir, our voice is coming clear and nice, sir.
Am I audible?
Yeah, you are audible, sir.
Okay. Okay. I can see all of you. Good morning to all of you.
मॉर्निंग सर
Dr. Ashok Kumar is not visible. Where is he?
He is coming. He is reaching.
Okay.
He was just checking here.
Good morning and welcome, sir. So, sir, I am waiting for the distinguished speaker, Dr. Barik, sir.
ज्वाइन कर लिया डॉक्टर बारिक ने
कहाँ है?
उन्हीं की ये सर मुझे लगा
डायरेक्टर
एक बार फोन करके पूछ लीजिए कि क्या ज्वाइन करने में कोई प्रॉब्लम तो नहीं है।
सीट में भी नहीं कर
मैं चेक करूँ।
कम टाइम में याद आएगा इसमें बंद करने के लिए बोला ना
क्योंकि वो इको होती है
ये हमारा म्यूट है
ठीक है।
टोन है
कविता
Good morning. Good morning, Dr. Barik.
Yeah, good morning, namaskar.
I'm T.R. Sharma.
Namaskar, yes. How are you?
Fine, thank you.
Good morning, everyone. Good morning.
Good morning, Barik, sir.
Good morning, Sokji, everybody. Yeah.
So, with the permission of chair, may I start, sir?
Yeah, please.
Very good morning to everyone. On behalf of NBPGR, a warm welcome to all of you to this webinar. As you are aware, on the occasion of the International Day on Biological Diversity, we are organizing this online program. This year's theme is Building a Shared Future for All Life. The theme was chosen by the United Nations to continue building momentum and support for the post-2020 Global Biodiversity Framework to be adopted in the upcoming Biodiversity Conference, that is the COP15. Our Honorable DDG Crop Sciences, Dr. T.R. Sharma ji, the chief guest for today, he has already joined. And our guest of honor, ADG Seeds, D.K. Yadava ji, will be shortly joining us. Today, we have the honor and privilege to listen to a lecture by none other than Dr. S.K. Barik, Director, CSIR, National Botanical Research Institute, Lucknow, who is a well-known scientist in the field of plant diversity, its conservation, impact of climate change on species and ecosystems. He would be speaking on the topic, Building a Shared Future for All Life through Utilizing Biodiversity Sustainably and Conserving the Threatened Elements. This is a topic which is very much in line with this year's theme. To begin the program, I invite Director NBPGR, Dr. Ashok Kumar, to kindly welcome the chief guest, the speaker, and the participants, and also to kindly introduce the speaker. Dr. Ashok Kumar.
Thank you, Dr. Kanta. With the permission of Chair, on behalf of NVPGR and my on personal behalf, I welcome our beloved Deputy Director General, Dr. T.I. Sharma ji. He has kindly agreed to be the chief guest of this function in the today morning at very short notice. So really, I am very thankful and grateful to you to be here during this morning. I am also thankful to Dr. and welcome to Dr. D.K. Adwa. He is out of station. He will be joining later, and he also agreed to be here in this morning. So I welcome to him also. Sir, on behalf of ICR or on behalf of the ICR NVPGR, I welcome Dr. S.K. Barik sir, Sarojkant Barik sir, the director, CSIR NVRI. He has kindly agreed to give the lecture that is very, very relevant to this year's theme. So on behalf of NVPGR, I welcome you, sir. You are, beside your busy schedule, you have kindly agreed to give the lecture for which you are very known to the biodiversity and including agrobiodiversity. So I welcome to you, sir, Barik sir. And I also welcome the Dr. S.K. Sharma, the former director of NVPGR. I can see directors like Dr. IIAB Ranchi, Dr. Subhash Chandra from NCPI. I can see here Dr. DOGR Pune, Dr. Major Singh, and many more scientists from the ICR system. And on behalf of NVPGR, I welcome all the ICR directors, the ICR staff, ICR Institute staff who have joined to this morning. And I also welcome to our own colleagues from NVPGR, Head of Division Station, administrative staff, and the technical staff of the NVPGR to this very good morning. And sir, regarding this International Biodiversity Day is being celebrated to increase the awareness among the biodiversity issues. So this year the CBD has the theme for this shared future life. As sir, you are kindly aware that the PG are the component of the biodiversity. So the conservation and sustainable utilization of the germ plant is the major mandate of the NVPGR. So sir, we are directly involved in the biodiversity conservation, biodiversity evaluation, utilization, and related issue to the biodiversity. Sir, with this, I would like to share a few things about the NVPGR. So during the last year, the NVPGR has involved in the conservation of the X2, but we have taken an initiative to develop the biological heritage site for the in situ conservation of the rice germ plant, that too in Assam. So with this initiative, we are fully involved, we are fully mandated for the conservation of the agrobiodiversity, particularly for the plant genetic resources, crop genetic resources. Besides the welcome matter, I have been also given the responsibility to introduce the distinguished speaker of the today, Dr. Barik sir. So on behalf of NVPGR, I would like to introduce Dr. Barik sir. Barik sir is a very towering personality in the field of biodiversity, agrobiodiversity. So I have, so it will take so much time. So I am in nutshell, I would like to say that at present, he is the director of the prestigious CSIR Institute, the National Botanical Science Institute, Lucknow. And before joining to CSIR, he was the head of department of botany at the botany department at North Eastern Hill University, Shillong. During past 3 decades, he has been actively involved in the plant science research, particularly the inventarization, bioprotection, conservation, ecological modeling, population, molecular ecology, chemical biology, carbon sequence, and and so many issues related to the ecosystem. Dr. Barik sir has successfully demonstrated the value addition in turmeric, production of turmeric leaf oil, oleoresin, value addition in the community level of the Meghalaya. Sir, he has published more than 150 research papers and edited so many books. Dr. Barik had selected as a lead fellow leadership in environment development International Rockefeller Foundation USA. He is the fellow of many prestigious societies. So he has the research specialization in bioresources, agriculture, forestry, ecology, environment, bioproduct development, and biomedical and healthcare intervention. Then he has significant achievements as a National Chief Technical Advisor. He has conceptualized implementing the Amrit Biodiversity Park around 300 acres, the part of the Central Vista. Leading the International Union of Biological Science, he is the member of 12 countries. He is the advisor of the Ministry of Environment, Forest. He is significantly contributed for the biodiversity development in the DBT. He has developed innovative methods for environmental flow, estimation, cumulative impact assessment, and determination of sustainability ecosystem. He is the mission director of major CSIR society missions, the CSIR technology mission, cotton mission, the CSIR plus mission, and food safety mission. He is the All India coordinator. He has implemented 10 research and development projects of DBT and the Ministry of Environment, Forest and Climate Change. As a PI, he has implemented 40 national, international projects for the industry and the research and development projects. He has filed 12 patents and published so many papers and reviewed books. He has commercialized 6 product technology and significantly contributed for the policy related to science and technology sector. Other than this, he is a fellow of many societies. He is a fellow of NASI, the NAS Fellowship, National Academy of Agriculture Sciences, Linear Society Fellowship, the Lead Fellowship, the R.N. Tandon Memorial Award. He is the Professor Viva Sahni Memorial Award of the Lucknow, then the Biodiversity Lecture Award. Besides, he has the professional responsibility. He is the Vice President of the National Academy of Sciences India, that is NASI. He is the board member of the SERB, the Department of Science and Technology. He is the member of governing body of the GB, National Institute of Government India under MOEF and Climate Change. He is the member governing body of Institute of NIBGR. He is the UGC nominated expert member, Center of Advanced Study in Botany, the BHU Varanasi. He is a member of Academic Council of Nehru and Punjab Central University and other 15 school board. He is the chairman of the Apex body of the Nehru Biotechnology Program in DBT Government of India. He is also the Technical Advisory Committee, the chairman of Technical Advisory Committee, and the expert member of 80 committees of Government of India, including so many committees. Besides this, I know he is also the member of the Research Advisory Committee of the Botanical Survey of India, and NVPI is quite lucky, fortunate to be as a member of the Research Advisory Committee of the NVPI. Sir, with this your short introduction, I am really thankful to giving a time to NVPI and sharing your view, your practical view on this very relevant theme of this year's. This is the theme of the Biological Diversity Day. With this, I again welcome to all of you, and I request to the chair to allow us for this lecture. Thank you, sir.
Thank you, sir. It is now time to hear from our eminent speaker. May I request today's speaker, Dr. Barik, to kindly begin his lecture.
My slides are visible.
Not yet, sir. Yeah, now they are visible.
Can you see it?
Yes, they are visible.
Yes, sir.
Please go ahead.
Okay. Thank you for inviting me. It's not moving.
We are looking at the picture forest canopy.
No, it is not moving. Okay. Respected Dr. T.R. Sharma, DDG Crop Sciences, ICR, the chief guest of the day, Dr. D.K. Yadav, Assistant Director General Seeds of ICR New Delhi, Dr. Ashok Kumar, and all the directors and scientists and ICR family and other participants, ladies and gentlemen. Let me first thank NBPGR, Dr. Ashok Kumar and Dr. Veena for inviting me to talk today on this day of one day before the World Biodiversity Day celebration, being a holiday on Sunday. So thank you so much for inviting me. Well, I just thought what topic to be given, so I took the pathway that what the global decision, the theme of this year's Biodiversity Day celebration that I included in my topic itself, building a shared future for all life. And of course, I added to that, how do you achieve this? Because we must utilize the biodiversity sustainably so that it is not overexploited, it is sustainably managed, and we leave something for our posterity. And conserving the threatened elements, that is the strategy through which we should do. So that's how the topic is. The title is a bit long, and we say mouthful of word, but I think it conveys the meaning of this year's World Biodiversity theme. Again, it is not moving. There is some problem.
Are you not able to go to the next slide, sir?
Yeah, now it has gone, but I don't know. In between, it is stopping. Okay. So as we know, let us see if it creates problem. I will reload again, but now it has come the first slide. Let me just briefly tell, as all of us we know, biodiversity, that is all the variety and variability of the life forms, not only on this earth, 3.5 kilometer above and 3.5 kilometer below this earth surface, the life exists. And that is the biodiversity. And fundamentals, all of us we know. So it is the ecosystem diversity, species diversity, genetic diversity, and another one is the cultural diversity, which cuts across the theme, all these three diversity levels. So when we talk about the ecosystem diversity, or in that matter, the genetic diversity, it's not only that structural diversity which we see, but also huge number of diversity or huge dimensions of diversity, which is attached to that functional diversity. So whether it is the molecular level, or it is at the ecosystem level, whether it is a nitrogen cycle where numbers of microorganisms are involved, a number of enzymes are accelerating the pace of decomposition or any other biological process. So all these functional diversity, they also constitute a part of the, whether it is ecosystem level we are talking, or it is the genetic level we are talking. So you will find that it is not the structural, but also the functional diversity is inbuilt within this diversity concept. Say, for example, when we talk about the ecosystem diversity, we are not talking about the variety and variability of the species that they contain, but also the varieties or diversity of habitats they contain. And on those habitats, you will find many of the species that are highly specialized. So you will find that, as I said, functional with the nutrient cycling or the regeneration processes or decomposition processes. So all the primary productivity. So there is a lot of diversity which is inbuilt in that. So we include those. So when we talk about the ecosystem diversity, we have huge number. If you see Champion and Seth, almost 218 diversity of ecosystems you will find in India itself, starting from the 0 meter elevation up to the, you can say 7,500 till you reach the permafrost. There is a diversity of ecosystem that ranges from tropical evergreen forest, which I showed in the previous slide, tropical evergreen forest, which is in the high rainfall area you find, and just see the diversity within just small area of around 100 meter by 100 meter you will find so many, and this is just 10 meter by 10 meter quadrant, and you can see that gigantic species. This is from Arunachal, Eastern Himalayas. And also with the secondary forest where the habitat is so bad. Here I would like to focus on the habitat diversity. Just see, this is the limestone area of Meghalaya on the border of Bangladesh, where there is a mining is going on, and see it's full of limestones, and on that also you can see the species are there. So this habitat diversity also creates a niche, specialized niche for the different components of the biodiversity. It is not only the plants, but also associated animals, microbes, and at every level. Well, you can see here, this is from the Lohit basin. You can see I have taken just a little from the top of the hill, and you can see that the tributaries of Brahmaputra, that one major tributary is Lohit, and you can see that this is the floodplain, how it looks like, and see that that gives the vegetation. Here is the mountain as you climb and up in the Himalayas. This is when we go from Gangtok to North Sikkim. On the way, you will find the mountain forest. It is a subtropical forest where you will find the diversity. Again, you can see the physiognomy of the forest, how it changes. Reach in the oak forest. Well, as you climb up and up, we have reached now the upper mountain forest above 3,500 meter elevation, where you will find the abyss densa forest is dominating. This is the origin of Tista River in Sikkim. Well, this is a very dense forest. You can see in the background that abyss densa forest, and in the foreground is the rhododendron forest, where at least 36 species in this particular forest exists. But in Northeast, you know, it is more than 83 species of rhododendron, including Arunachal and Sikkim. So this is from Sikkim. So you will find the origin of Tista. You can see here. So abyss densa stand, yak is grazing. Most of the time it is snow clad, but it is a summer season photograph. So you can see that water is flowing. Yes, this is a glimpse of the rhododendron diversity. It's a single species, and you see that the diversity, this is from Sikkim, different colors. Now we have reached the Alpine Meadows, where the tree line is ending, and we have the very beautiful grasslands, and you will find that, you know, this is the time when you travel the area, April 15 and to May 15, every 15 days you will find that, yes, potentilla, they keep on changing their colors. Today you are seeing yellow. After 7 days or 10 days, if you go, it will be all purple. Then you will find all pink. Then it is all red. So that is the beauty of our Alpine Meadows, and which is the grazing ground for the yak. Yes, we don't have only the diversity in the terrestrial ecosystems, which I showed you a glimpse, but we have also interesting ecosystems. This is near Dhubri. When you cross the Brahmaputra, this is the river island. So no electricity, but you will find the life is thriving there. And this is from boat I took it. This is again another story of like all of you know this. Fundis, that is also a typical ecosystem, and you know the life forms there. So it is not that ecosystem diversity, but the life forms or the diversity within those ecosystems also emerging. And in country like our India, you can see the diversity. Also, we have the coastal ecosystems, and you will see these are the diversity. If you see, they also possess a lot of functional diversity, as I said. Now let us come to the, you see, for example, here it is not only the species diversity. When we talk, it is not only the higher plant species, but also the microbial diversity. And you know that it is very little understood, little studied because of the culture method, the bottleneck. Now with the 16S RNA metagenomics approach, we are now able to move very fast. And you can see this is an example which we are studying legumes and rhizobia diversity in the Northeast. And in the beginning, we studied all these inventory, but now we are going to the functional characterization, which is a major project from DBT we have started now, that what are those molecular mechanisms that decide that a particular legume will be highly specific to a particular rhizobia, be it the brady, be it the cynorhizobia, but they are very highly strain specific. But to our surprise, when we're doing some manipulative experiments, we found that one particular legume, which is a specific rhizobia symbiont, when we introduced in a completely different habitat and climate in the Rajasthan from Meghalaya, we found that a new symbiont has come in. So that means the specificity of the host and the symbiont relationship is not highly specific as we were thinking before. So in order to unravel that mechanism, now we are understanding that what are those molecular mechanisms, whether it is the just simply what we know now, NIP gene or node factors, or it is the elicitors or effectors, or it is the transporters, or it is the metabolomics of the rhizospheric zone, or it is the miRNA or MIPAPs. So what is those experiment, those molecular mechanisms, signaling mechanisms that work? So you see, just even at the molecular level of functional diversity is so vast. So this is an example, human rhizobia diversity is so vast that itself. Yeah, so when we studied the, thanks to the metagenomics approach, so 16S rRNA gene targeting that, we could identify many new and unique species which we are now working on that more rigorously. Well, and you can see here the glasshouse tree draining pot experiments were the authentication and cross inoculation studies of rhizobia. So you know that whether a particular species is specific to a particular symbiont, or some of these symbionts which have the very beneficial, whether it is the phosphorus solubilizing activity, or it is the nitrogen fixing ability, or they have the multiple functions. So how do they impact the cross species inoculation studies for the rhizobia, so that they can also be useful for our rice or other major crops? So that type of experiments we do. So you see the diversity, not only the genes, but also the miRNA diversity. So for example, MIR 160 is extensively important for the nodule formation and start. So you can see the different levels of the nodule formation process, the different miRNA are playing the key role, or it could be also MIPAPs also. So this is the progress project which we are doing now, the completely unraveling the mechanism, so that I hope that that diversity varies from the each and every one, from one legume to other legume, it varies. So we are trying to unravel that story. So the diversity, just see that what level of diversity at how many layers it is functioning. And this is the Indian recorded species and world record. These are all common statistics, but I must tell you that now at least BSI has finalized that angiosperm diversity is almost 21,000 plus. 21,000 exactly, 21,400 something. So this is the species within the species. So Rhiza sativa, you can see the varieties. Thus this claims there are many, and India is, you know that it is the origin. Well, along with all these three layers which I discussed, the ecosystem, genetic, and the species diversity. So that is the rice was the example of the genetic diversity. And along with that, the large number of functional diversity which are associated. We have the cultural diversity which cuts across this theme. And because of our culture, many of our species, many of our genes, many of our molecules in the process, they are conserved. Otherwise, they would have vanished by this time. These are the factors which are really threat to our biodiversity, whether it is the ecosystem level, as you can see from a forested landscape, it is completely converted to a mine landscape, or here the sand mining is going on and the whole forest will go. And you can see the shifting cultivation in Northeast, where I have spent my life 30 years. You can see that how the nicely covered, dense forested landscapes are covering into the simply bare and completely nuded hilltops. So the shifting cultivation. So we must do the conservation, not that we are not doing. Conservation is important, as you know that ex situ and in situ conservation measures are always followed. So thanks that, okay, we cannot say that it is all 100% fulfilled, but substantial initiative has been taken by the government since 1975 or 76. You can see that we have today 106 national parks. 106 national parks, 564 wildlife sanctuary. It is a huge number, 670 protected areas. That means total, it is a good success, and more than 5% of our geographical area is under the in situ conservation area. It is not that only the protected areas, that means wildlife sanctuaries and national parks, but also we have 18 biosphere reserves. So we have 99 conservation reserves, 218 community reserves, 51 tiger reserves, 18 biosphere reserves, 32 elephant reserves, 49 sites for wetland international Ramsar sites. So like that, we have 467 international bird areas. So several conservation initiatives have been made, whether it is the ex situ conservation in the botanical gardens, all these things we have done it. Yet we are not able to conserve our species, not only in India, but global problem it has. You will see that the number of species being extinct, it is a huge. So we say 10 species per day and the estimate varies, but fact is that yes, we are losing our species in a very fast rate. I will not talk all those fundamentals, but this much is enough to say, yes, 6th mega extinction event is on. And nobody else, it is not that external factors like meteorites or other factors. It is the human anthropogenic cause is the cause of our 6th mega extinction event, which is already ongoing. Unless, because we are the culprit, we are responsible for destruction of these species, pushing them to the faster extinction. That's why it is our responsibility to really conserve these species. Otherwise, okay, as you know, background extinction will always go on. But yes, because we are accelerating the pace of extinction, it is our moral and ethical responsibility. We have to conserve them. So that's how the discipline of conservation biology actually was born. So it is, we say, mission-oriented crisis discipline. Truly, it is an ICU. If we don't work intensively and urgently, nobody can save, whether it is the species or genetic diversity or ecosystem diversity. What I said, the activities are so intense. It is the scale that matters. So that's why we say conservation biology is the mission-oriented crisis discipline. Yes, Earth is on the ICU and we have to save it. The discipline is tied closely to ecology in searching the dispersal, migration, demographics, effective population size, inbreeding, depletion, and minimum population viability of certain species. Yet, conservation biology is an interdisciplinary subject, not only drawing ecology, genetics, evolution, biology, and biotechnology, but also in social science, economics, and conservation policy. So together we can really do the justice. The principles underlying each of these disciplines have immediate implications for the management of species and ecosystems, captive breeding and introduction, genetic analysis, and restoration. So interdisciplinarity is the need. Well, J.L. Harper, nature conservation has changed from idealistic philosophy, a little bit background about the conservation biology. So you will see that plant resource management goal is largely utilitarian or commercial. But yes, nature is worth conserving rests on the several fundamental arguments, particularly the argument of nostalgia and the argument human benefit and need. People argue that yes, since the natural process, it is the natural selection, whoever wins, they will be surviving. Why we should be bothered? So that's what I said. It could be the argument could be nostalgic that yes, no, we must save our neighbors on this Earth surface, but also we use them for our own benefit. So that's why conservation is essential. So the most important part is that is unfortunate thing. We have been talking so, but you know that even today in India, 33% of our geographical area, at least botanically, is unexplored. Forget about the microbes, hardly 1% or 2%, even after the metagenomic technique available, then also we have not crossed beyond 2%. So that means the title of this year is that you think about others who are sharing this earth with you. But unfortunate thing is you do not know who are they. So is it not the same for the scientific community? Even after the independence, we have not been able to completely explore our neighbors on this earth surface. Okay, India has the problem of the human resources or it is the financial resources. But yes, I think we must prioritize that. Unless we know who is there, forget about what for they are. By prospection, I will talk at length subsequently. That is, forget about that. At least we must know who are there. Inventory must be completed. Well, so the tribute to Michael, this gentleman is known as the father of the conservation biology. Michael is soul. Unfortunately, during the COVID, we lost him last year, 2020, June 17. So he is the father. We say that he sought to bridge a gap existing at that time between theory in ecology and population biology on the one hand, and a conservation policy and practice on the other. Theory we are studying, but practice was not happening. Credit goes to him. Methods have been developed to address fundamental problems in conservation biology, from the most traditional forms of conservation to new approaches at genetic to landscape scales, consequently showing how the science can be put into practice. So we salute him and we pay our tribute because he is no more with us. Recently, he left us 2 years back. Conservation toolbox to translate conservation theory into practice. Metapopulation modeling and population viability analysis, which I will show you today. PVA for comparing the relative effects of potential management actions and population growth, or persistent to determine the minimum population viable size, MVP, the specific probability of reaching the extinction. All these things I will demonstrate today you so that my objective is not to just give a talk. I would like to see that each and every institution, each and every citizen takes care of, learns these methods and start conserving. If one species we can save, that will be great. We don't have the luxury of spending $8 million per species like, bald headed eagle of USA. We are a poor country. So unless we make it a people's movement, each and every one of us become responsible and do scientific conservation, I think the rate at which the extinction is taking place, we cannot save our species. We will keep on talking that we are a mega biodiversity rich country, but yes, they are really dwindling in a very, very fast rate. Well, conservation genomics, from conservation genetics to genomics, some of these things I will discuss today. From conservation genetics to conservation genomics, I will not talk more details on this, but yes, now the time has come and we are working on the conservation genomics. So it is not that genomics is only for functional genomics or variety development, but also it can be successfully used in the conservation aspect. Once we know the detailed total genomic composition, it is very easy to manage them. So we are now working on the conservation genomics as well. Earlier, we were emphasizing the conservation genetics, which we have used in our project. Well, the extinction depth and colonization credit, some of these concepts in the conservation biology, I must say that yes, the time lag between a particular extinction process starts and the real impact it starts, that is the extinction depth, or earlier it was relaxation time. But yes, we also have the opportunity to create new habitat and patches for colonization. So that's why we say that colonization credit. So where the corollary is the extinction depth is a negative phenomenon, where colonization patches availability, or that is the colonization credit, is a positive phenomenon. So while planning the conservation planning, the area for this, particularly the second colonization credit, we must earmark that. So these are the very minute level thinking process when we do the conservation planning. Yes, climate change integrated conservation strategy, this is a must today. Climate change integrated conservation strategy can be a good starting point to understand the complexities of the conservation biology under a changing climate. Relatively few continent-wide extinction of species have been documented during abrupt changes in the past. So, for example, Pleistocene, a sign that species are probably more resilient to climate change than anticipated in model assessments of the effects. Based upon the past records, of course, the extinction rate is high, but when we talk about the climate change vis-a-vis conservation, at times we feel that models are underestimating or overestimating the number of extinction. So that part we have to check it. Biodiversity hotspot and cold spots. We all know that there are 36 biodiversity hotspots on this Earth surface, but cold spots might be a little bit new. So many of these people today, many of the scientists that are arguing, no, we must define the cold spots as well. That means those areas, as you know that wherever the 1,500 endemic species are there, and at least 70% of those original areas have been destroyed, those are the biodiversity hotspots. All of us we know. And in India, we have 4 biodiversity hotspots out of those 36 global biodiversity hotspots. But what is this cold spots? Cold spots means when we identify the biodiversity hotspots, our resources, our attention, our efforts are all concentrated toward those 36 biodiversity hotspots. And we leave the remaining part of the world. And that is the cold spot. So with less investment, with less effort, these cold spots also they have high diversity, we must not forget. So that is the latest argument. Well, we started, say for example, in India, this is the Mosia robusta when we started this mega DBT project in the year 2009. So we had only one species of this one individual of this one in the bank Tinali, or Mosia robusta, and that was also subsequently cut down. Fortunately, by that time, we regenerated it vegetatively and we saved this species. This is an endemic species. Nowhere on this earth you will find other than that. So only one tree was left, but fortunately when we started searching them, we have now at least 6 individuals we have found in different places. They are far off places. So that is also an interesting mechanism to study. Where is Pasighat, where is Itanagar, and what is Balfakram in Meghalaya? But you will find one on isolated or 2 trees are there, total 6 tree species. At least we are happy that instead of we thought that one species has been extinct. So how do we search these individuals, species, or the cryptic species concepts, all those things are there, but how do we search? Gone are the days when we were moving with a vehicle and putting there and then start walking 100 kilometers, 200 kilometers to search a species. But today is you can see, of course, these are threatened category species. We are using ecological niche modeling, which helps us in getting the species, and those predicted areas we go and search. In the process, you will find that we have increased the number of new populations, and accordingly, through that modeling only, we could know this species presence there. So now modern tools we have to use, and some of those I will see. And of course we know that now, please don't use the word RET, rare, endangered, and threatened. That is the American, you can say, the Endangered Species Act terminology, but I think today if we use it creates confusion. IUCN is the globally accepted terminology. So here the endangered is a category of the threatened. So threatened category itself is an umbrella term under which you can see critically endangered, endangered, and vulnerable. These are the three categories. So these are quantifiable parameters, and we have the methods for that, and we are now classifying them. So thanks to the Department of Biotechnology, as I said, from 2009 to 2017, we run this project, which I was coordinating, 37 institutions, 150 scientists across the country. And we could, at least I could say out of 100, we targeted 76 or 77 species. We have successfully conserved it. And this is a special issue we brought out in the current science series on the cover page, Conservation of Threatened Plants of India, where the detailed methods have been described. So you can see this is the one of the chapters, geographical distribution pattern of threatened plants of India and steps taken for their conservation. So if where you completely reviewed it, what has been done so far, this is a chapter in that, you can see it, February 10, 2018 issue of current science. So total we have compiled the list of 2,704 threatened species which we have got in India. But please remember that this is based upon the conventional classification, herbarium-based classification, not population-based classification. So those 2,704 threatened plants, we have found now many of them that don't deserve to be in the red list. They should be brought out, because when we had a detailed survey now, we found that they are not really threatened category based upon the IUCN criteria. So very quantified ones. So also many species are there which have not got a place in this red data book, which they need to be brought in. So it is natural. Any science when we do, not that time when it was classified, that time it was important. Whatever tools, techniques available, scientists have done it, but it is the time we must revise that based upon the more realistic and more advanced techniques available in our hand. So you can see this 2,704 could be plus minus 200 or 300. That is the things, and you can see that they comprise 2,641 angiosperms, 23 gymnosperms, 44 pteridophytes, and Orchidaceae is the dominant one. And you can see the other families are Fabaceae, Poaceae, and Rubiaceae. Co-dominant families are Asteraceae, Euphorbiaceae, etc. But most important is you will find that 408 genera were represented by 2 to 9 species, while 582 genera were represented by 1 species each. So the most major trends are less than 1% of the threatened species had wide distribution range, indicated by their presence in 10 to 23 states of India. So less than 1% of the threatened species. That means 99% of the threatened species, they are restricted to the small 1 or 2 or 3 states, and you will find that 62% of the listed threatened plants of India had restricted distribution to only 1 state. So that means each one of us, they have the responsibility, because a person from Lucknow or from Shillong or from Delhi cannot go and take care of a plant in Andaman. So each and every country, and they constitute 62% of our threatened species. So there is an inconsistency in the threatened status report by the different sources, as RDB, Redrabu, IUCN, Sastrinaya, or IUCN, or NBS, or NBA, Biodiversity Authorities, or state notifications, CAMP, that is, and Schedule 6 of Wildlife Protection Act, and also CITES appendix. So you will find there is a lot of inconsistencies, classificational error. So that's why we took it, and you can see that this is the summary of those things earlier classified. But now we took up these 10-point protocols, so I will not waste time. But I must say that we followed population inventory, characterization, and mapping using ecological niche modeling. That saves your time and accuracy. Instead of blindly searching, we can really give it. And that's how we did it first for the bird flu virus, and after that it was in the cover page. Then we found that in the plants also it is very successful, and we have demonstrated, and throughout the country we have trained people on this technique. Metapopulation modeling to determine the conservation status, minimum viable population size, and assess the extinction risk. Earlier, what we were thinking, just an arbitrary, how many trees you would plant so that it is become out of red data book or something, conservation status is improved. But yes, metapopulation modeling will give you the exact number, how many you should introduce to get a particular improvement in the conservation status. So that also we have trained people throughout the country, I hope. And many of the papers are coming up now, so we are happy for that. Identification of factors responsible for depleting species populations and developing a species-specific recovery strategy. just because something you have to do science, a species doesn't have any reproductive bottleneck, but it is being uprooted, overexploited. So if you do the reproductive biology for that species, it's useless. We use less wastage of money, wastage of time. So you understand first what for this species is being threatened. What is the cause? If it is a management issue, give it to the forest department, let them manage. If it is grazing, so let them do. But the science part, the scientists should do, like the bottlenecks, or say molecular characterization to identify populations with greater diversity for genetic enrichment base. That is the sourcing concept. So if a particular, as you know, genetic drift will start if the genetic base is very low, narrow. So in order to avoid the genetic drift, we must see that which is the genetically diverse population, and from that we can use, that can be used as a source, and in the genetically impoverished population that can be a sink, and slowly you can increase. We tried this in case of two species, in fact, we tried, and the Aconitum in high Himalayas, we tried this, but still that experiment is going on. Yes, it will enrich, but generation after generation you have to monitor. So these are the 100 certain plants which you tried, thanks to the joint effort of 150 young and established scientists, and there is a whole group team which I was coordinating. But these are the 100 lists of species, and I'm sure that 76 of these species are well, because follow-up also we take. So that is now at least the populations are perpetuating. Well, these are the species, but so the Department of Biotechnology so far we have taken, and it is a meager, very less number of species you can see. So something, not all the aspects, some aspects have been taken care of, and you can see that number of, so it is a very meager number. We have to increase it manifold. So this is the example of the one Ilex khasiana, where the IUCN reported only 7 individuals. When we utilized the ENM, we found that it is 3,070 individuals are there. So that's how it uses the correct figure, and you can see that this is the example how ENM gives you, and this is the utilization. This is in the current science paper throughout the country. You can see in Jammu Kashmir some of the species, and Northeast India, for example, we have given it. So when we did this experiment, this study completely, and you can see the IUCN classification, many of the species you know that they have not assessed at all. And many of the species you can see, say for example, Ilex venelosa was endangered, and we also confirm it is endangered. But our confirmation is based upon, you see so many parameters. You know that A, B, C, D classification in the IUCN, and of that you can see the B methods we have used here, and C whatever method data available. And say Rajasthan immunity carpa was endangered, but we found it is critically endangered. So it is, once you assess it, you can see for here is the Vesta robusta. It was least concerned, but we found it is the critically endangered. See, just at least it is 3 steps ahead of the conservation status. So it is, that's why I said that always work with the population. And it is, you see, Acer Hooker was not assessed at all. When we assess it is critically endangered, the highest conservation status. So it is high time that all of us you collect the data for the population, and IUCN criteria we did it. So you can see the important centers of plant species richness using it is a citrus study we did it, the ENM application. Also the identifying corridors for landscape connectivity. So you know that for the wildlife we always do the corridor connectivity. For plants we have developed this model how it can be connected. Modeling hotspots for invasion alien plants. This I can say you, this paper, this particular work was actually important because we found that all the biodiversity hotspots, all the biodiversity rich areas are also hotspots for the invasion. And this is the largest immunity carpa study. You can see the distribution around 137 distribution points, and around 200 individuals are only there. So we did that. So this is a study again we did it. That means distribution models usefulness. In government of Ecuador called us to really see that which are the areas where it can be suitable. They want to replace the paddy with the lotus, and they wanted to use our Indian lotus, that is the Nucifera. So we went and we identified their areas completely in Ecuador where this can be suitable, and we are supposed to give them our Indian lotus with high nutrient capability so that they will have a high export value. And it is, we are also trying to propagate this. And when you do this work, we developed many new species. You can see when you do the species search, all your MVPGR people all are doing, but I will advise that if you are searching a particular species, go for the ecological niche modeling, and you will find the cryptic species part, you will get many of them. So these are the new species during this modeling. because they have the overlapping niches. So naturally, in the areas where the particular species the model has predicted, you will also get the new species. So that's how we got the 7 new species, and these are some of the examples: Paridasanai, Tawangensis, and so on and so forth. And this is the metapopulation model, where as I was telling, what I was telling, that you can directly, precisely predict how many years this particular population or particular species will take for extinction. So this is the M1 and M2 scenario. M2 is the undisturbed and M1 is the disturbed. So naturally, in the disturbed habitat, you find extinction rate is very high. So the extinction probability will, or the persistence probability will be very low. So here you can see that M2 model scenario. So the probability model is phyla. So we have done for 4 species. So you can see here how many individuals to be introduced. So in the disturbed M1 model, you will find that more number of individuals to be introduced. And you see, as you increase the number of individuals here, you will see that the status from the least concerned to near threatened, vulnerable, say for these are. So we will now take it up and up so that they are out of the threatened category. Well, we took up all the methods we followed, whether it is the tissue culture rest or it is the simply cuttings, seed stem cuttings, leaf tip cuttings. So all the methods we followed, seeds, whether it is the high-end filarias or it is the Jaisalmer, throughout the country we took up, and that is the success story. Well, that is not adequate, because we have to see that this biodiversity should be put to use for a bioeconomy. As you know, bioeconomy is the future of the world. So if we are sitting on the biodiversity rich area, we must utilize them and sustainably utilize them, so that the population is maintained in the wild. We can cultivate them so that the pressure on the wild populations that is reduced. At the same time, we get benefit out of that, so that those countries who have the high biodiversity, there is probability, if subject to that we put our efforts, we get it. So this is an example how we did it. This is the RB1, RG1, this is the ginseng. Always we see in the plains, I came to know after only coming to Lucknow that there's an Indian ginseng. What is that? No, Withania somnifera. But actually we have ginseng in India. People do not know that. 7 species of ginseng, that means Panax, we have. And when I started working, this was from 2008, we started working, and you will see that these are the diversity of the ginseng. And one of them, you will see that one of them, particularly Asamicus, that is equal ginsenoside content as with the American or Korean. So why not popularize those? So once you do the bioprospection, many doors open. So this is the example, all this Hplc profile, we did it and we find that our species is there. But yes, difficult to propagate, but yes, it is not important. So this is the bioprospection is important. So you can see that, you can see here example, it is, you see the RG1 and RB1, both you see Meghalaya, this is Asamicus. So it is very high, 3.12. It is comparable with the Korean and American ginseng. Well, these are the Hplc protocols. So for example, Xanthoxylum aromatum, for this, as I said, I was working in Northeast. So, but is there any good scientifically proven mechanism of action, safety, toxicity, in vitro and in vivo, then finally release it through after the clinical trial? So that is a problem which you don't know. That's why China is ahead of us. So taking that as a challenge, those things, bioprospection which we did in the past, we started developing the products. So for example, here for the Xanthoxylum, you know that berberine and xanthoplanin and dictamine, they are the major components. So using that, we develop here NBRI Dentogel. And it really kills pain immediately, and it is really in their demand. We have commercialized it. And we have worked out all the mechanism of action, how it works as an anti-inflammatory, antimicrobial. So say for example, this is a Gualtheria fragmentissima. Everybody in the traditional knowledge they use for pain killing. So now we know that salicylic acid is the dominant one. So methyl salicylate, targeting that, and 1.25% of essential oil it has. So we developed the, now it is in the already 80% in the development process is over. So anti-arthritic drug we are developing. So for example, cytokine enzymes, COX-2, we target receptors TL4, we target transcriptional factors, NF-κB, we target analgesic. So targeting those things, which are it is working, so that we can biomarkers we can identify and we can work ahead. So chrysophenol, one of this thing, we're working on a, particularly our colleagues were working on the one lichen, and from there we isolated the chrysophenol. And when this was silver nanoparticles, we functionalized it. And when we found that it is very useful in the catheter, we put it on the catheter, and in catheter, it is completely antimicrobial. No microbial. Otherwise, earlier what is happening? Every 2 days you have to change the catheter, and by that time also the this antibiotic resistant microbes, they colonize on this, and it causes disease also to the patient. So now this particular isophenol functionalized silver non-particle coated one is now 28 days it goes. Till then there is no microbial infection. So all this testing has been done, and finally you will see that now it is in the clinical trial. So these are some of the things. I just give an example. Follow the scientific validation methods, both systematically, whether it is the IUS or it is the phytopharma mode, and use your biodiversity. This is an example for the, I just gave you healthcare sector. Say all the testings have been done, whether it is the fluorescent or everything. So finally we release it in the after that model. We go for the things. So you can see the next one example I'll give you, cannabis. Cannabis indica, all of us we know bhang, but it is an untouchable plant because that has so many medicinal uses, you can see. Particularly epileptic treatment, obesity treatment. So you know that, we know that, but because it is an untouchable, so we started work on this, and NBRI is the leader in this exercise, which I think we started in the 2016, immediately I joined here. We started on the cannabinoids. You can see that a class of chemical compounds, active constituents of cannabis that act on cannabinoid receptors. 120 plus cannabinoid types are there that have been isolated from the cannabis. And you can see the tetrahydrocannabinol. This is the culprit, actually, psychotropic substance, THC, shortly. And cannabidiol is the very non-psychoactive cannabinoid. This one, whatever you take, there will be no NOSA. But yes, it has many medical values, as I showed you in that list. So our target is, you know that CB1, that is the THC works on this in the brain. So this is the cannabinoid receptor CB1 for THC. But when we talk about the CBD, CB2 receptor works where the spleen, bone, skin, immune system. So it helps in treating several diseases. So the cannabis indica or cannabis sativa, cannabis indica is our own. But even if it is our own plant, but we cannot touch it because it has been a property substance. People are getting arrested, as you know. So THC, CBD. So now we are trying to develop. You can see here there are major components: CBD, CBD5. All these things are cannabinoid and tetrahydrocannabinol acid, THCA, CBC, cannabinoid, and all these are the compounds which we are targeting for the treatment of the different diseases. So not established for OTC products as yet. Only the one medicine, that is the Epidiolex by US FDA, they have approved it. So now we have at least 3 medicines are in the pipeline, following different methods, and these are the targets which we are working now. Our group here in NBRI: anti-obesity, appetite stimulating, pain relieving, anti-stress, lipstick, anti-epilepsy, and all these in vivo models which we have targeted, and in vitro models we are targeting. And you see, as I said, we are following in fact both IUS and phytopharma. Phytopharma, if we do it, then US FDA, it is possible to release those medicines also outside the country. So this is the way of how we are going. And here is the target for the anti-epilepsy drug development. Epilepsy is a very major problem, and here you can see that these are the targets which we are different proteins, coupled receptors, and what are those potential variants. So biodiversity, how to utilize scientifically so that we can really capture the world market as China has done today. So this is the appetite regulation. So that is again useful CBD. And these are the targets for the pain relieving, which are the receptors like TLR4 and of course B38 and MAPK, protein kinases. And here is the example for the obesity drug development. You can see that there are lipolysis, thermogenesis, adipogenesis, appetite, lipase. What I am trying to say you, unless you scientifically target the biomarkers you identify and scientifically you validate, nobody is going to accept your product. It is easy that everybody is doing, even jodi buti ka bhi ho jata hai. So you have to do it scientifically validation, and only when it is really found in the clinical trial very effective, then only you should leave it to the market and we will have a global presence. That is the biggest drawback in Indian system of medicine. Well, I will just now go to say the ecosystem level. This is a myrin sacred group where in Meghalaya we have huge number, but only 1% are left now. And it is a lot of ecosystem services. Say, for example, this paddy field you can see, for last 30 years, I have been seeing that not a single cow dung is put. Forget about the chemical fertilizer, they never use. So you can see that this particular land, this agriculture field continuously gets nutrients from this forest, and it is the bumper yield it gives. So that is the benefit, the nutrient cycling benefit of the ecosystem, one of the ecosystem services. So we map those ecosystem services, and you can see that 8 ecosystem services, whether it's the carbon capture here or biodiversity, and finally we map that these are the areas, these green areas are the in watershed level, micro level we planned, and these are the green areas where most of the ecosystem services are available. So these areas must be protected, conserved. So also we use the remote sensing, that as I said, techniques we use. So this is the example how plant richness pattern was correlated with the functional photosynthetic active radiation, so that we can really cut short the process and times and quickly get the result, how the richness is going in which trend. So like that, remote sensing, whether it is the direct, monitoring the shifting cultivation areas or the biodiversity loss, everything we monitor now, utilizing the very high resolution imageries, including the hyperspectral, and of course now still it is developing in the radar-based technologies. Well, this is an example where you can see that the local resources biodiversity can be utilized to improve the livelihood of the people. So you can see here, we started a model development in Tripura. As I said, in the German we are working. There is a 4-tier model actually we developed. One is the, the tree canopy in the top. Then the middle tier is the fruits. We do it fruit tree planting, and then the turmeric, ginger, or the rice, wheat we cultivated, and the below ground also tapioca, or it is the our papayas and all the colocasia, they are there. And that is the 4 model. But we found that although the income, the people, and of course planting the nutrient, nitrogen rich plants, as we all do always in the agroforestry system or in the different farming models, we did it. But one thing we found, there is a deficit, that yes, for the consistent or the permanent cultivation, from shifting cultivation to permanent cultivation transition, we found that nutrient level is going down. So biofertilizer is a must. So that realization led to us, and we developed this model now. We utilizing the biodiversity in each state through a NMHS project from a Minister of Environment Forest, we did it. Here you can see that we isolated from cultivation sites where you will find that whether it is the phosphorus solubilizing, or it is the nitrogen fixers, or it is the biostimulants for different purposes. We isolated ourselves and did it. And finally, we developed the biofertilizers following the methods of the CIV registration also, because some of them were bacillus. So finally, you can see that all those methods we followed. These are the code names for the bacterial things, and we finally molecular characterized also, identified them also based upon that, and we treated them right from the seed germination stage, and we validated that yes, they are doing the good performance in this rice crop. Similarly, we did for different other vegetable crops, and finally we developed the, you can say, following the biofertilizer production. You can see we trained also people and how to really generate, and you can see that these are the different methods followed, and we followed all the guidelines of the VIS, phosphorus soluble bacteria, for example, for each of them. And finally, the biofertilizer developed, and also trained people to do that. And all these things now in the field, people started applying them, and we got excellent result. And today we can say that yes, it can be now applied in the large scale. So that for different biofertilizers, so for different states, we developed the different, I got the different strengths and developed the biofertilizer for that. I hope now the shifting cultivation areas which were under permanent cultivation, the nutrients were depleting. Now the application of the local consortia biofertilizers, if really the methods are followed strictly, I think that problem can be addressed. Similarly, so this is for the different crops, we did it. And similarly, this is another problem you can see. This is the acid mine drainage in Meghalaya and Assam. We did it when I was in Nehu, and we found that yes, the problem is acid mine drainage pH is less than 2 at times, and you will see that how to do that. So we isolated the bacteria from those areas only. And of course, this is Assam, that is Meghalaya. So after those isolation, we did it heavy metal screening also tolerant bacteria, because that is not only the acid mine drainage, but heavy metals were also there, iron, manganese, and lead. So then we identified them through the 16S rRNA. Then of course they were used both anaerobic and aerobic chambers we established, very small, because this was we are targeting, as you know, small miners are there in Meghalaya. They cannot invest large money for the environmental rehabilitation. So we use the local plants for the mining areas to rehabilitate, and for the AMD treatment, we isolated the local microbes who have the capacity to really neutralize the AMD, along with the both passive and active methods we used. limestone and the bacterial composition, both anaerobic and aerobic chambers we differentiated, and water was allowed to pass through that. So this is the model which we developed for the small miners, and total cost was around 50,000 rupees. So this is the example you can see. So the local resources we used, whether it is the hydro or it is the microbials, accordingly you will see that the water outlet from the lateral mine, now it is almost 6 point something the pH now. So the large areas now we have started treating them. And this is the last story I am going to tell you, that many of those important resources which we have, we are always proud of, but yes, they are depleting. The one story is like this: unless you give them compensation, unless you give them appropriate price, nobody is going to cultivate those things. So utilization, conservation through utilization, but unless the farmers get adequate compensation, why they should do. This is the story of Lacadang turmeric, all of you know, which is now geographically tagged. GI tag is there. But the issue is now that 20 rupees a kilo. Who will cultivate this one? So I thought that from DBT I got a support from the DBT from the Bioresource project. We developed this infrastructure. And this is the turmeric, Lacadang turmeric. As you know, the people said that varied numbers, but I have done in my lab myself, and 13% I got it. Even now also in Odisha, we cultivated this, it is the 9% curcumin. So it is really a very rich curcumin rich. Of course, it varies from one place to other place. When you plant the same plant down below in Nangpo, it becomes 6%. So, and also in Lucknow, when I planted, it is coming around 4.5 or 5, something like that, depending upon the place. So the issue is it is a high curcumin content, but why? Only 20 kilometer radius this one grows, where you will get 13% of the curcumin. So this one we took off. So unless you add the value, it cannot be done. So I started this program with the women self-help groups. Around 127 group people were involved. We covered around 200 hectares area, and you can see that this is the turmeric from where oleoresin is extracted. After that, also this particular powder which is left now, this is also 3.2% curcumin still left after taking oleoresin out, from where we take up the curcumin and also the bioavailable curcumin now we are developing, which is useful. As you know, 99% of the curcumin which you take, it is not available biologically. So we have made it biological. This is the technology from NPRI, we developed it. And now this is the model. You can see this is the plant we established in, sorry. No, it's not going. There's some problem. Anyway, so this is the story of turmeric. After that, you will find there is the socioeconomic contribution is there. So we must use it judiciously, the biodiversity, the value addition, the total picture, somehow it got lost. All the components are also there. Slicer, boiler, everything we established from the DBT support. It was not a very big amount. It was around a few lakhs rupees, but I tried to connect with the other departments, particularly that time IPAD project was going on. So they gave the building. Somehow this is the building, but the total component wise it is not visible. 2 places we established. So where you will find the big pulverizers we put up, and 8 hot air oven dryers with 52 trays each. So completely mechanized we did it, and that is a good experience. And here you can see there are 4 vessels are there, 1, 2, 3, 4. This is the leaf oil. The leaf oil of turmeric is completely different from the rhizome oil. So leaf oil, we have a product also now in NBRI. After those oils, we are developing one floor map when it was. But this is all the other 3 are the oleoresin extraction vessels. So the economics is very good. Even in the small, this is a pilot scale plant where capacity was 1500 kg per day, and then the raw materials. So it has economical benefit per day. They were adding at least 2 lakh something, so that it is quite beneficial. So that is my point. The technology which we work, we must go to the people. They must reach people for their benefit. Laboratory scale, we do many things, but whatever we do, it must reach people. Then that it is true also for the conservation. So it is, you can see that you are utilizing, but through cultivation. So we have identified the germplasm, elite germplasm, then both economical benefit, also the species is naturally conserved. The area which was shrinking, now it is enhanced actually. Not only in Meghalaya, now at least in 30 square kilometer and 30 kilometer radius, it is now being cultivated, area is increasing, but also we are trying in other states also. So for example, as I said, in Koraput, in Odisha, or in Maharashtra, we have given them, and we are now trying to see that there are 6%, 9%, although the curcumin content is varying, but it is nevertheless the local variety which is 3 or 4% of the curcumin, certainly it is better. So in the process, this Lacadam variety is now well conserved. So that should be a model for many of the resources which I've said. I thought that I must present the stories of my own life experience which I have with my colleagues, because as you know, this type of activities we do with a group, and many of the people have contributed to this. I cannot list them individually, but I thank each one of them for making all these stories possible. And yes, if I means my talking to you, it will be successful if any one of you or all of you actually initiate some action, whether it is directly planting or identifying the certain species or utilizing some of the valuable resources into converting them into bioeconomy. I think that is my objective, which I have been doing, and I thought that more people, it is always better. And yes, utilizing the bioresources, biodiversity should be utilized and it should be converted to bioresources, and bioresources should be converted to the bioeconomy. And that is the future of the country. Thank you so much once again for inviting me. And I am fortunate that my friend and Dr. P.R. Sharma, who is the DDG, he is the chief guest today, and under his chairmanship I am talking today. And all of you, the whole ICR family who are listening to me, and I will be happy if you have any question. Thank you so much.
Thank you, sir. Thank you very much, sir. We thank you very profusely for your wonderful lecture. You extensively covered the topic with beautiful examples drawn from your own experience and your own stories, which you told very, very nicely. So, are we going to take any questions? Audience? Does the audience have any questions? If so,
It's okay, let's go ahead then. Yeah.
Yes, okay. Thank you, sir. Thank you very much. Moving on with the program. Now I request Dr. Sunil Archer to give a brief about the debate competition that we had organized on the occasion of the International Biodiversity Day. And I would also request him to announce its winners. Dr. Sunil.
Yeah. Thank you very much. Good afternoon, everybody. Wonderful lecture indeed. Thank you very much, Professor Barik. We all know that the Convention on Biological Diversity is in force since 29 December 1993, and 196 nations have ratified the CBD. And after around 20 years, the Nagoya Protocol has also come into force, and 128 countries have ratified so far. Similarly, International Treaty on Plant Genetic Resources for Food and Agriculture, ITPGRFA, is in force since 29 June 2004, where around 149 nations have ratified. These multilateral agreements aim to ensure biodiversity conservation and to link benefit sharing to the sustainable use. However, on one hand, advancements in technologies such as genomics and gene editing have brought a new paradigm to how traits of genetic resources can be accessed, allowing what is called as dematerialized use of biological resources. And on the other hand, benefit sharing obligation being optional or voluntary has not helped the cause. The question therefore is whether the carefully constructed multilateral systems have achieved their objectives at all. Should they be abandoned altogether? 4 teams debated on are the multilateral international instruments of biodiversity conservation successful in building a shared future. Each team had one speaker arguing for and the other against the motion. We had 4 teams, and the teams comprised of Dr. Suma from Regional Station Trishul and Dr. Sherry from Division of Germplasm Conservation in the first team. Second team, Dr. D.P. Wankhede and Dr. Yasin Jashima from Division of Genomic Resources here in Delhi. Third team, Dr. Neelam Shekhawat and Mrs. Chanchal Jaina from Regional Station Jodhpur. And the fourth team comprised of Dr. Badal Singh and Dr. Harish, GD from Division of Germplasm Conservation. We had two learned judges having expertise and experience in the PGR policy, Dr. Sanjeev Saxena and Dr. Pratibha Brahmi. Participants had 5 minutes to make their points and 2 additional minutes at the end to debate on the other points. At the end of the debate, based on the content, style, and confidence, judges have decided that team having Dr. Sherry and Dr. Suma as the winner and team having Dr. Badal and Harish as runner-up. So we congratulate them. In fact, we congratulate all the participants. And the judges have also have asked us to mention the appreciations to Dr. Neelam Shekhawat for her excellent participation. It was an excellent experience, wonderful experience for all of us. I hope everybody else has also enjoyed. I thank the debaters as well as the audience for making the experience pleasant. Thank you very much.
I congratulate all the winners and thank you, Dr. Sunil, for giving us a brief about the debate competition. Dr. Yadava has joined us now. Welcome, sir. I now request him to Dr. Yadava to kindly give his remarks.
Thank you, Dr. Kavita. Thank you very much. And good noon to all. Honorable DDG Crop Sciences, our Dr. T.R. Sharma ji. Today's outstanding speaker, Dr. S.K. Barik, Director NBRI. My director colleagues, Dr. Ashok Kumar from NBPGR, I see Dr. Patnaik from IAB Ranchi, Dr. Lakshmikant from Almoda, Dr. H.V. Singh from NBIM, Bau, and Dr. Major Singh from Directorate of Oil, Onion and Garlic from Pune. Dr. S.K. Sharma, our Honorable former Director NBPGR, he is also seen on the screen, and all our head of the divisions, head of regional stations, our scientist colleagues, students, and all staff. So my hearty congratulations on this day of this Biodiversity Day, and my compliments to the organizers for selecting such a wonderful speaker for today's presentation. And I personally congratulate Dr. Barik also that he has put the exhaustive picture of the biodiversity globally in context to Indian context. And whatsoever is spoken, I think I could attend only half of the lecture, but based on his own expertise, based on his own work which he has done. So he has put a very good picture. So here what we could understand that he has tried to tell that where we were earlier, what we are today, and where we are to go, how to go further using the various tools for the conserving our biodiversity, particularly where the various species which are being threatened. So in that direction, he has made a very good attempt. And I think the take-home message from what Dr. Barik has presented, that to save the life on the globe, I think the saving of biodiversity is essential. And in his talk, I think he has taken all the value addition and nutraceutical parts of and how the in the NEH region biodiversity they have revived and how they are working in that. So thank you very much, sir, for very nice, I think, work you have presented, and that is very, I think, valuable for all the participants, our scientists and our students also. Our ICR is also, I think, working very extensively on the collection, conservation, characterization, its sustainable use, and the sharing the germplasm amongst all the National Agriculture Research System and the other organizations who are involved in the crop improvement program. And for that, already our 6 bureaus, they are working. The most important one where we are sitting, we are working, this talk has been delivered, who has organized this program, our National Bureau of Plant Genetic Resources, then our Animal Genetic Resources, Fish Genetic Resources, the agriculturally important microorganisms at MAU, and then this insect resources at Bengaluru, and then National Bureau of Soil Survey and Land Use Planning at Nagpur. So I think all our 6 bureaus, they are taking care of all the diversity available in India, and they try to make available whatsoever is available globally, so that our scientists can exploit that particular genetic diversity in any of the species. And our bureau, actually, it is a very proud moment that we stand at number 2 globally, where we are conserving more than 4.5 lakhs of the various accessions with of more than 2,000 species at our bureau. And as far as this traditional varieties and the crop wild relatives are concerned, so more than 96,000 that accessions, they are also being maintained by us. And I think this material is being shared very frequently, and all the efforts are being made to the endangered species, that how we can conserve them, how we can avoid any danger to that. So those all aspects we are taking care of through the various projects, in-house projects, also in collaboration with DBT also, and through one of the ICR's project on CRP on biofortification. So all the bureaus, they are working together under this program, and I think a very good work is being undertaken by all our colleagues here. So I once again compliment all our participants, and particularly our speaker, Dr. S.K. Barik, and my sincere thanks to our Honorable DDG Crop Sciences for being with us during this program. So thank you very much to organizers also for giving me this opportunity. Thank you very much.
Thank you, sir. Thank you very much for your kind remarks, and also for being a big support to NBPGR. It is now my honor to invite our DDG Crop Sciences, Dr. T.R. Sharma, to give his address.
Very good afternoon to all. Am I audible? Am I audible?
Yes, sir. You are audible, so please go ahead.
Yes, sir. Yes, sir.
Good afternoon to all. My very good friend, Dr. S.K. Barik, Director, NBRI, Lucknow. Dr. D.K. Yadav, colleague from ICAR ATG Seeds. Dr. Ashok Kumar, Director, NBPGR, New Delhi. Dr. S.K. Sharma, former Director and Vice Chancellor, HPKVB, Palampur. Dr. Distinguished Directors of different institutes of ICAR, staff of NBPGR, ladies and gentlemen, I would like to congratulate you all on International Day on Biological Diversity, NBPGR, for organizing this very important talk by an eminent speaker and expert by Dr. Barik. The talk on building a shared future for all life through utilizing biodiversity sustainably and conserving the threatened elements. He has very eloquently explained about ecosystem diversity, forest diversity, starting from tropical to temperate to subtropical, legumes and rhizobia diversity, conservation biology, conservation of genetics and genomic resources, conservation of threatened plants, then conservation using different methods, and very eloquently explained about how biodiversity can be converted into bioeconomy by using the principles of Ayush, phytoplasma, etc. And then ending his talk very nicely, giving examples of development of rural technology incubation center and utilization of bioresources to bioeconomy. So it was a very comprehensive talk. I really enjoyed listening to Dr. Barik's presentation. And that was a very good justification for our institute NBPGR for the celebration of International Day for Biological Diversity today, which is going to be celebrated in fact on 22nd May. So congratulations to all the organizers, and also thanks to Dr. Barik for sparing his valuable time to be here this morning and delivering a very comprehensive talk. We know, friends, that the United Nations has proclaimed May 22nd as the International Day of Biological Diversity to increase understanding and awareness of biodiversity issues. And we know that in December 2000, the UN General Assembly adopted 22nd May as International Day for Biological Diversity to commemorate the adoption of the text of Convention on 22nd May 1992 by the Nairobi Final Act of the Conference for the adoption of the agreed text of Convention on Biological Diversity. This is a very important occasion, and that is why the Secretariat of the Convention on Biological Diversity has announced the Biodiversity Day 2022 slogan as building a shared future for all life. The slogan was chosen to continue building momentum and support for the post-2020 Global Biodiversity Framework to be adopted at the upcoming UN Biodiversity Conference, that is on COP15. We know that biodiversity remains the answer to several sustainable development changes, from the nature-based solutions to climate, health issues, food and water security, and sustainable livelihood. Biodiversity is the foundation upon which we can build back better. The Convention on Biological Diversity will adopt a post-2020 global diversity framework as a stepping stone towards the 2050 vision of living in harmony with nature. And that is very interesting to note that even the slogan for this year's Biodiversity Day is building a shared future for all life. So it tells us that we have to live in harmony with the nature and give space to all living organisms while taking the benefit from each other and have a good symbiotic relationship with other organisms. Mention was made by Dr. Yadav on agrobiodiversity. I would like to say that our institutes, all these 6 bureaus which were mentioned, are the custodian of agrobiodiversity in India, starting from micro to plant to animals. Therefore, we should understand that it's very important to conserve, characterize, and utilize biodiversity with very cautiously, and also giving space to all living organisms and then making benefit out of the biological diversity surrounding to us. We know that our planet is filled with agrobiodiversity, is very rich in agrobiodiversity, and out of 4 lakh plant species, at least half of them, around 2 lakhs, are edible for human consumption. Around 6,000 species are used as a food, and 150 crops are cultivated on a significant we deal with 85 crops in the Indian
Others please video off, then his bandwidth will increase.
And out
If
of
others off the video, then his bandwidth.
only 3 crops dominate food.
Sir, you are not audible, sir. Didi साहब आवाज नहीं आ रही सर आवाज.
We know that. I will try to switch over.
हाँ सर अभी आई है थोड़े वीडियो ऑफ करके सर बोलेंगे सर ठीक आएगा सर.
Am I audible now?
हाँ yes sir.
अब ठीक हो गया.
So we know that India is a very rich gene center and one of the 12 world mega biodiversity. One of the 8 centers of origin of crop plants is also present in India. Parts of 4 of the 35 hotspots of biodiversity existed in India, which consist of Himalayas, Indo-Burma, Western Ghat, and Sundarland. So about 29% of the flowering plants are endemic to India, and 19,000 species of higher plants are present in Indian subcontinent. It's a very rich source for more than 385 crop species which are cultivated, and 817 native crop plants and wild relatives are also present in Indian subcontinent. And 1,000 naturally occurring edible plant species are present here. Therefore, large-scale ex situ collections and is very, very important, and our National Bureau of Plant Genetic Resources is playing an important role in ex situ conservation as well as in situ conservation of plant genetic resources. And we know that these plant genetic resources are very important for UN Sustainable Development Goal and meeting the challenges of these Sustainable Development Goals. Out of those 17 goals, 6 goals, that is number 1, no poverty, number 2, 0 hunger, number 3, good health and well-being, number 12, number 13, and number 15, life on land. All these 5 goals are directly or indirectly related to plant genetic resources, and we must therefore conserve, characterize, and utilize plant genetic resources as well as biological resources of other organisms, so that we can make use of them in our food system. It's a very, indeed, a matter of very satisfaction and pleasure for me that our institute, our bureaus have made efforts to lead to the notification of wild rice biodiversity heritage site at Barzoli on 7th May 2022 by the government of Assam. And ICR NBPGR believes that it is extremely important to
It seems that there has been a power failure in the area, and our Honorable DDG Saab is unable to join again. So let me continue. I would like to thank him for his kind support, his inspiring words and motivation. Thank you. At the end of this very educative and interesting lecture and the wonderful remarks of our guests, I would like to propose the formal vote of thanks. First of all, we wish to express our gratitude to the Honorable Dr. T.R. Sharma, the DDG Crop Science, for chairing today's session. Thank you for readily agreeing to be the chief guest and for his keen interest in biodiversity and PGR. We are extremely thankful to Dr. S.K. Barik, Director, NBRI Lucknow, who is a renowned scientist with a vast experience of more than 30 years, for his wonderful lecture. He covered the topic very, very exhaustively and packed a lot of information in 1 hour. We are extremely thankful for that. And it is indeed our good fortune that we got the opportunity to listen to you, sir, on the occasion of the International Biodiversity Day. We also thank Dr. D.K. Yadav for his kind and gracious presence. Although he was traveling, he was able to join us. Very special thank you to you, sir. I also want to thank our guests from other institutes, our former director, Dr. S.K. Sharma, who was here a couple of days back. And when he came to know that we were having a lecture by Dr. Barik, he immediately said, please send me a link. So his presence is itself a blessing for us. Thank you, sir, for joining. A special thanks to our director, NBPGR, Dr. Ashok Kumar, who has been involved in right from the beginning and for steering the organization of this entire program. I also thank my colleagues, Dr. Veena Gupta and Dr. Sunil Archak, for organizing the debates, competition, and to all the teams for their active participation. I thank my colleagues, Mr. Vijay Mandal, Ms. Kanchan, for their help with the logistics and invitations. A special thanks to all my colleagues in the PME cell, Dr. Rajkumar, Mr. Arun, Vikas, and Sanjeev for all their help and support. A very big thank you to all the participants. I can see that a lot of people from other institutions, from our regional stations and staff have joined. Plus, we have also given the YouTube link, so we don't know how many have joined there. So a lot of people have joined. I wish to thank each and every one of them for joining us today. At the end, I wish to thank one and all who have contributed to making this program a big success. Thank you.
Thank you. Thank you. Bye.
Thank you.
Your lecture will be available on YouTube, sir.
Sharma ji is still so interested.
ये तुमने उस टाइम
भूल गए?
शेयर क्यों नहीं कर रहे थे?
नहीं, यहाँ कहाँ है वो, उधर से शेयर कर।
अच्छा वो उधर बता